Let's get ready to review for our Unit 3 test. Unit 3 Graphic Organizer class review. You should have gotten a handout before starting this video. Please make sure that you have it. If you're watching at home, there's a link on your checklist so that you can print out this organizer as we go through Unit 3. Now, first we're going to start out with our passive transport part of our chart. We know that passive transport goes from high to low. It is natural. There's no energy needed. Passive, low energy, you're not really doing anything. Things go naturally. People want space. Molecules want space. Again, we go from, remember the elevator example where if I crowd 10 people in an elevator, 5 people leave the elevator, you don't stay in your own spot. You sort of move around to fill up the space because people don't like being in their bubble. Same thing with cooking. The highest concentration of smell is in the kitchen, and over time, the smell spreads out because that's the air molecules that have the smell, again, attached to it, is spreading out throughout the house. And so you can smell it further and further away the longer the time is. And eventually it dissipates because it's so spread out. So again, things naturally go from high to low. Now you have 3 types that we needed to know. You have the first one where I want you to look at this image. It's first just natural, and this goes from high to low. Super simple, main type. We call this diffusion. And this is the image that you're going to need to know. So make sure that you're paying attention to these images. These images will be on your test. Next, let's identify the other one. This main, again, hint is going to be water. Also, high to low. So we also know it needs no energy. And the last one, we have a couple hints. Big, high to low, and protein. So it's diffusion, but it's diffusion with some help. And they need proteins. And the difference for why they need help is they need that protein to help. So if you look at the image, you're still going from high to low in your image, but you have these big green proteins that are helping you. Now let's talk about how we will know, either from the definition or from the image. So does it need energy? Is it going from low to high or high to low? And in this case, we know that diffusion needs all molecules. So let's answer that question. So first off, doesn't need energy, goes from high to low, and all molecules do this. Gas, water, whatever. Now, osmosis is going to be pretty much the same. We probably realize that the need energy and high to low is going to be the same for all of them, but our big hint is going to be water. I want you to underline water. Water is your big hint. And I'll go on and tell you, you're never going to have an image of this because we didn't give you this image in your notes, but you do need to know the definition. Now, facilitated diffusion, again, does not need energy. Again, high to low. But the next part is that we see big molecules, and it uses a big molecules and proteins in the definition. And then we also see the protein in the image. Now, this one is going to be similar to protein pump. People confuse facilitated diffusion and protein pump a lot because they both use proteins and they both use big molecules, but the difference is one needs energy, the other one does not. So you've got to remember, facilitated diffusion is diffusion with a little bit of help. We know diffusion needs no energy. Let's go on to our next one. So active transport. We know that active transport, activity needs energy. This one, we're packing things in, low to high. It's not natural, and it does need energy. Let's take a look at our chart. So in the same way, we're going to identify, we have one type, and then we have bulk transport, which there's 2 examples of bulk transport. So this one, we just talked about it, uses proteins, goes from low to high, and it needs energy. This is a protein pump. It's pumping. It's using proteins to pump something from low to high, and it needs energy. Again, if you look at the image, low to high, and to go in that, you need energy. Also look at the image, ATP, energy. You can actually see it. Now let's go to bulk transport. One type of bulk transport goes from the inside to the outside. So it's leaving, it's exiting, and it needs energy. The other one goes from outside to inside. It's going inside. It needs energy. So we have exocytosis and endocytosis. Now let's look at the images. For protein pump, again, you see that big protein, in this case it's purple. You're going from low to high, you see energy. In exocytosis and endocytosis, exo exiting, you see a group of molecules exiting the cell. In endocytosis, you see a group of molecules entering the cell. Now let's go on and fill out our how do we know. We know that protein pumps need energy. They go from high to low, or sorry, low to high, and you're going to see a protein. Exocytosis needs energy. It's going to go from low to high. It's a group of molecules exiting, exiting. Endocytosis needs energy. Yes, it does. Low to high. It's a group of molecules entering the cell. All right, moving on. Okay, so we're going to talk about this big term that we associate with this, and it also explains why our cells do certain things. But the main thing is we know that passive transport and active transport help the body maintain homeostasis. And this is both the internal and external balance of the body. So if you think body, homeostasis. So make sure you write down body and homeostasis in your notes. So these points help us maintain that. They also help us with getting nutrients for our cell energy, which we'll be talking about in lesson 2 and 3. But before we leave it, we got to talk about tonicity. Let's practice some tonicity. With these problems, if you follow this step by step, you are going to get them right each time, but you've got to follow the step by step. All right. So the first thing that we always do in a tonicity problem is we count. We count the outside, then we count the inside. So let's do that with this first one. We count the outside, count the inside. We have 6 and 9. 6, again, all we care about is the solution right now. So we're labeling the solution. The solution has the low count, so we're writing low. And we know hypo low. So that's going to tell us that we have a hypotonic solution. And that means that the water is going to actually rush in. So we have arrows that are drawing water rushing in, and the cell's going to swell. In a hypotonic solution, cell's going to swell because the water's going to rush in. Now, next one. Let's count. Outside's going to be 7. Inside's going to be 3. The 7 is the high one. Again, we were paying attention to the outside of the cell, the solution, not what's inside the cell. We know hyper high, hypertonic. And we know that in hypertonic, that means our water is going to be pulled out. So water's going to rush out, which means our cell is going to shrink. Now let's back up real quick to hypotonic. This is one of those things that if we have a solution with this, if we have an animal cell, we have a possibility of that cell actually bursting. Whereas a plant cell, this is really important because they're always in a hypotonic, a lot of times solution when they're especially pulling in water. You're going to not see that because they have the cell wall that protects it. Now let's jump to the last one. We're going to count our 2. We have 5 same, and then we have 5 on the outside. So those are the same. Isotonic. Iso means same. So we know that as one molecule moves in, one molecule is going to move out. So we have water molecules moving in and out, which means our cell will stay the same. Now let's jump to lesson 2 and 3. Now we're going to review these together because we've studied them separately, but now you're going to have questions that are going to link them together and you don't want to get confused. So we need to study them together. Don't solo study. In other words, don't only study lesson 2 and only lessons 3 separately. Study them together, mix them together. We're at this point. Now, we have photosynthesis and cellular respiration. They both are processes that help deal with energy. Now, the first thing we need to know about photosynthesis is autotrophs, producers, plants. These are the organisms that do this. They're the only things that can be out in the sun and go, give me energy. Whereas cellular respiration, all living things do this. Plants and animals do this. Because all living things need to take glucose, break it down, and turn it into ATP, which cells use. Now, the definition for photosynthesis, it is a process that converts energy from the sun into chemical energy to be stored for later. Cellular respiration is a process that releases energy from food, aka glucose, to turn it into energy cells can use, which is ATP. Again, all living things do this. Plants do this, and animals do this. So plants, they make the glucose, but sometimes their cells need that glucose. So the plant is always during the day is going to be making that glucose and storing it up. Remember, it's a larger amount of energy. And then when the cells need it, It takes the glucose, breaks it down using cellular respiration, and turns it into energy that the plants can use. Now, we know we need this because we can't get energy directly from the sun. So, a heterotroph, which is all of our animals, we have to do this process. Now, when do these occur? Photosynthesis, because it is reliant on light, it has to get to sort of start the process, the light dependent reaction. You're going to find that it's most active during the day. Now, cell respiration, it's always going on because guess what? Cells always need energy. Now, where is this occurring? We're talking about the big ticket organelles, not so much what's in them. So, photosynthesis happens in the chloroplast. Now, the light dependent reaction specifically happens in the thylakoid of the chloroplast. You do need to know this. And the Calvin cycle happens in the stroma. Now, in cell respiration, we have two organelles, but mitochondria is still the main one because it makes the main amount of energy that we need. Cytoplasm just does glycolysis and fermentation, and that's just a small part. The biggest part of cell respiration, the main part, happens in the mitochondria. So cytoplasm for glycolysis and fermentation, mitochondria from Krebs cycle and etc. And when we get further down into our charts on the next page, you'll notice that there are specific locations in the mitochondria. Krebs cycle happens in the matrix, and etc. happens in the inner mitochondrial membrane. Now, with this equation, you've got to understand what's on the reactant side and what's on the product side. You'll notice that they're flip-flopped. So what one starts with, the other one, and what one starts with and then makes, the other one starts with what was made and then ends up with what was started. The only difference is that we label the type of small energy differently. We call sunlight, because really though, when we capture that sunlight, it turns in, it's small energy, it's ATP. And in cell respiration, we don't deal with sunlight at all, so we can't use that word. We just use ATP. But you've got to know that if you have to have those coefficients that are in front, like you know that you need 6 molecules of carbon dioxide, 6 molecules of water, and sunlight. If you don't have all of that, if you just have 5, photosynthesis can occur. And it is going to make glucose and 6 molecules of oxygen. With cell respiration, we know we need a molecule of glucose and 6 molecules of oxygen to be able to start it. Once we have that amount, then we can continue onward with creating the ATP, the carbon dioxide, and the water. Again, those are 6 molecules, each of those. Now, we know that sometimes organisms don't have enough oxygen. And in that case, we do have something that happens without. So we'll come back to that in just a second. Let's jump over to our photosynthesis additional specifics. Now, we know that only autotrophs do this, and that chlorophyll is a pigment in chloroplast that captures that light energy. That's definitely going to be a question. Now, let's jump back over here. We know autotrophs and heterotrophs do cell respiration. We know that with O2, with oxygen, we go glycolysis, Krebs cycle, etc. But without oxygen, we go to glycolysis, and then we go to a fermentation. Depending on what the organism is, that depends on what type of fermentation. If it's bacteria or human, lactic acid. If it's yeast or fungi, it's alcoholic. And again, these are very small amounts of energy. So in other words, they take a glucose molecule and they don't get a lot out of it. So it's not exactly what humans like to do. We only do it during, again, emergencies. Now, let's talk about some extra concepts. We know that we're going to need to be able to identify these images. They were on our quizzes. We need to know this. We need to know that that first image is ADP, and we're adding energy into it and adding a phosphate. So we're storing it, and we're storing it to make ATP. The other one, you can see we have those 3 phosphates for the ATP, but we break off one, and that is going to release energy. So we know that forming bonds stores energy. This is going from ADP to ATP, and that breaking those bonds, that phosphate off to set it down to 2, is going to release the energy. We also need to know, we use inputs and outputs later on, but we need to know that interchangeably, reactants is what goes into a reaction. In other words, that's the input, what goes in, and products is what leaves it, what's at the end, what's produced. Now, you have these 2 images in your packet. You're going to see these images again. I've labeled on them letters. you could use your last page that's blank on your graphic organizer to create a key using your notes, because we completed that, and to be able to answer, because you're going to have a couple questions from each of these. Now let's talk about the lesson 2 and 3 steps and locations. We're going to start off with just photosynthesis in general. We'll go with a light dependent reaction and then go to the Calvin cycle. So photosynthesis, we are pretty much just taking sunlight and we're making glucose. Now, light dependent reaction, the big summary for this one is that we're taking that chlorophyll is going to capture the sunlight and use it to charge up ATP and NADH into small short-term energy. Remember that ATP is small and short-term. Now, the inputs, what we need, the reaction of what we need, the reactants of the light-dependent reaction is we need sunlight and water. We're going to have some ADP and NADP plus that's coming from the Calvin cycle after it's been sort of drained. They're going to come back and cycle back. So we know that they're there because we're going to be making ATP and NADPH. Now, our output, our product is we produce oxygen. The plants, not we, but the plant produces oxygen. The plant doesn't need it. It's a byproduct. Its main goal is to make ATP and NADPH. And those ATP and NADH then move over to the Calvin cycle in the stroma. Again, remember, light dependent happens in the thylakoid. Calvin cycle happens in the stroma. And we're going to take that ATP, NADPH, and the carbon cycle that it takes in. Remember, that's the one thing that we hadn't used yet. We used sunlight and water, hadn't used carbon dioxide yet. And we're going to create glucose. This is a large, long-term energy source that's going to be used for storage. Again, carbon dioxide is going to be, again, the base of the glucose with all that carbon and oxygen already there. And then we're going to, again, take the energy stored in the ATP and NADPH, break those bonds, release the energy, charge up a glucose so that we get glucose. And then we have some ADP and NADP plus that have been used up, and they're going to go back to the light dependent reaction like we talked with our input. Now let's talk about cellular respiration. With cellular respiration, we want to make sure that we understand that we're starting with one glucose molecule. And we're going to get 2 plus 2 plus 32 equals 36 ATP. So if you're asked how much one glucose molecule will make, it's 36 ATP. Now let's talk about glycolysis location. Glycolysis happens in the cytoplasm. And what's going to happen is it's going to take that glucose, that one molecule of glucose, and it's going to break it into pyruvic acid. And it's going to do that. It's going to, again, take that glucose, break it into pyruvic acid. Now, additionally, it's also going to produce some ATP and NADH. Please write those down in your notes. Those are numbers. Again, the ATP is just the general stuff that's being produced. The cell's going to use those. NADH is going to go eventually to the ETC. Now, that pyruvic acid that was just made in glycolysis is going to enter the mitochondria. It's going to go to the mitochondria matrix, where Krebs cycle is, and it's going to be broken into carbon dioxide. And again, Krebs cycle needs its reactants are pyruvic acid, ADP and NADP plus, and FAD plus, because those are items that are going to be charged up when they break the pyruvic acid. Remember, breaking bonds releases energy and allows that energy to be stored up in other things. Now, the output is that we get carbon dioxide. Again, not needed, but it's a byproduct. We create it, again, by breaking down that pyruvic acid, and then we get what we really need, which is the ATP, the NADHs, and the FADH2s. Make sure you write down those numbers. Remember, the NADH and the FADH2s are going to join the NADHs from the glycolysis and going to the ETC next. Last phase, we have the electron transport chain. We're going to go from the matrix to the inner mitochondrial membrane. And in this case, we're taking the NADH and the FADH2 from the previous steps, and we're breaking those hydrogens from them. And then we're taking the hydrogens and we're putting them together with the oxygen. That's where we get the oxygen that we breathe in. It's needed to, again, have something to take those hydrogen ions and add them to. And it's going to allow us to charge up a lot of ATP by breaking all of those bonds. So we take 10 NADHs, 2 FADH2s, remember those came from glycolysis and the Krebs cycle, and then the oxygen from the atmosphere that we breathe. We then charge up that ADP to make a lot of ATPs, 32 to be specific, and we also make water. So in the ETC, we take those hydrogen ions, those electron-carrying items that we add to oxygen, to a molecule we come from the atmosphere, and we get water. That was probably a question that you might have gotten right, hopefully, on your quiz, but if not, hopefully you won't get it wrong on your test. So let's look at everything. We need to take a holistic, again, look at this material, because if we study and to get too intricate, if it gets too intricate, it's going to be hard for us to remember. So a couple things that we want to end with. To study, take this information and rewrite it in a different way. I took our notes, I summarized it. I didn't write everything word for word. I broke it down, and I also broke it down and rewrote it differently, because that made my brain think about how I wanted to organize it. And as I organized it on my paper or on this document, I then was able to organize it in my brain. You can do that too. You're just going to rewrite it differently. Now, as you write, think about the ways that you can remember the information and keep it straight. It may be silly, but if it works for you, great. Think about how we remember the difference between hyper and hypo. Hyper high, hypo low. Again, can be silly, but if it makes sense to you, do it. You can even make a review game out of it. But whatever you do, do not just reread. It does not work for you. There's too much information here. It's too complex. You're not going to be able to keep it right. You've got to work with it. This is not going to work. You're going to set yourself up for failure if all you do is just look at your notes. You've got to do something with it. You've got to make a proof, even if you don't get correction points for it, because again, we're in the test right there. We don't do corrections for that. Now, remember that we're also going to study in small amounts. You don't want to sit down and study for 3 straight hours for this. You need to take breaks, and you also want to switch from different subjects to different lessons and things like that. So maybe study lesson 1 for 25 minutes. Take a break. You may not be finished with lesson 1, but that's okay. You're going to come back to it. Start on focusing on lesson 2 and 3. Maybe focus on the big differences between the two. Take a break. Come back, study for 25 minutes. Maybe study this specific part. Again, what's going in, what's going out, trying to keep it straight. Take a break. Maybe go back to lesson 1 and study a little bit more of that one. And again, as you return, switch to different lessons. You want to practice harder than you ever imagine you'll have to for your test. The less prepared you are, in other words, if you've not done any studying, if you've not worked with your study guide, if all you've done is study a little bit for just your quizzes and that's it, you've got a lot to do. Hopefully I'll see you in the afternoon review sessions, because again, studying with someone can make it go a lot better. Good luck. Make sure that your graphic organizer is filled out. Have fun playing the review games, and hopefully I'll see you either Tuesday or Wednesday for our review session.